Hi, my name is Sarita, lecturer in botany, GDCW Nalgonda. Today I am going to discuss about structure of anther. It is also called microsporangium. Anther is also known as a microsporangium. This topic comes under embryology. This topic belongs to the embryology. Embryology is one of the unit of third semester of second year BSc BJC. Embryology is one of the our topic of embryology is the one of the unit of third semester of second year BSc BJC. Before going to discuss about structure of anther, we have to know about embryology definition. So, I want to give brief introduction to the embryology. The study of embryos and development of embryos is known as embryology. The study of embryos and development of embryos is known as embryology. Father of Indian plant embryology is Acharya Panchanan Maheshwari. Father of Indian plant embryology is Acharya Panchanan Maheshwari. Panchanan Maheshwari was prominent Indian botanist noted for his invention of the technique of test tube fertilization of angiosperms. Panchanan Maheshwari discovered the technique of test tube fertilization of angiosperms. This invention has allowed the creation of new hybrid plants. This invention created the creation of new hybrid plants. He also classified plants according to the difference he found in such embryological studies. So, he was called father of Indian plant embryology. Panchanan Maheshwari wrote about an introduction to the embryology of angiosperms, and he is also editor of recent advances in embryology of angiosperms. This is about Panchanan Maheshwari embryology definition and Panchanan Maheshwari, the writer, the father of embryology. Next, coming to the our topic, structure of anther. In angiosperms, the male reproductive structure is called stamens. In angiosperms, the male reproductive structures is called stamens. The stamens have filament and anther. The stamens, these are the stamens. The stamens have mainly two parts. They are, this one is filament, next anther. The stamens have mainly two parts. They are filament and second one is anther. The anther is present at the end of the filament. This one is anther. This anther is present at the end of the filament. This anther is nothing but male reproductive structure. This anther contains male gametes which are called pollen grains. In angiosperms, the male reproductive structure is called stamens. These are the male reproductive structures. These are called stamens. Stamens mainly have two parts. They are filament. This one is filament. Second one is anther. The anther is present at the end of the filament. This anther is nothing but male reproductive structure. Anther contains male gametes which are called pollen grains. This anther is based on the lobes. This anther is two types based on the lobes. This anther is two types. They are dithecous anther and monothecous anther based on the lobes. This anther is two types. They are diathecous anther and monothecous anther. Anther having two lobes. This one is one lobe. This one is one lobe. This is one lobe. This is one lobe. This is another lobe. Anther having two lobes is two thecus is called diathecous anther. It is found in many plants. It is found in many plants. In diathecous anther, the septal wall is found which divides the anther into two lobes. Anther is known as two lobes. Anther having anther having two lobes is called diathecous anther. Anther having this one is one lobe, this one is another lobe. Anther having two lobes is called diathecous anther. It is found in many plants. In diathecous anther, the septal wall divides the anther into four lobes. This septal wall divides the anther into four lobes or four foliar sacs. In diathecous anther, anther having two lobes is called diathecous anther. Anther having two lobes is called diathecous anther. It is found in many plants. In diathecous anther, the septal wall divides the anther into four lobes. This one is one. These are the four lobes or four polar sacs. Next, anther having only one lobe is called monothecous anther. Anther having only one lobe is called monothecous anther. This is found in the family of Malvaceae. This is monothecous anther is found Malvaceae family. Only one septal wall divides the anther into two lobes or two pollen sacs. Based on the lobes, anthers are two types. They are diathecous anther and monothecous anther. Based on the lobes, anthers are two types. They are diathecous anthers and monothecous anther. Anther having two lobes is called diathecous anther. It is found in many plants. In diathecous anther, the septal wall separates the anther into four lobes or four pollen sacs. And coming to the monothecous anther, anther having only one lobe is called monothecous anther. This is found in Malvaceae family. Only one septal wall separates the anther into two lobes or two pollen sacs. This is about diathecous and monothecous anther type. Anther is known as microsporangium. This anther is known as microsporangium and ovule is known as megasporangium. Anther is known as microsporangium and ovule is known as megasporangium. Next, coming to the our topic structure of anther. Before going to discuss about structure of anther, we have to know about the development of microsporangium. Development of anther or microsporangium. Coming to the development of anther or microsporangia. Microsporangia development starts from mass of primary sporogenous cells. Mass of primary sporogenous cells takes periclinal division and produce outer side parietal cells and inner side spore mother cells. Coming to the development of anther or development of microsporangia, a mass of primary sporogenous cells takes periclinal division. Periclinal division means in the group of The group of cells is called tissue, contains upper surface and lower surface. In this periclinal division, the division takes place parallel to the upper surface and lower surface is known as periclinal division. Periclin. Primary sporogenous cells takes periclinal division. Periclinal division means a group of cells. A group of cells are called tissue, contains upper surface and lower surface. Division takes place parallel to the upper surface and lower surface is known as a periclinal division. Primary sporogenous cells takes periclinal division and produce outer side parietal cells or primary wall layers and inner side spore mother cells. Parietal cells again take periclinal division and produce sporangial wall or anther wall. This anther wall contains epidermis, endothelium, middle layer, tapetum. Next, coming to the inner spore mother cells. Inner spore mother cells take meiotic division and produce haploid microspore which are called pollen grains. Once again, we see development of anther. Primary sporogenous cells takes periclinal division. A mass of primary sporogenous cells takes periclinal division. Periclinal division means a group of cells called tissue having upper surface and lower surface. In this periclinal division takes place parallel to the upper surface and lower surface is known as a periclinal division. After periclinal division, it produces outer side parietal cells and inner side spore mother cells. Parietal cell or primary wall layer again takes periclinal division and produce anther wall or microsporangia wall. This wall contains 4 layers. They are epidermis, endothelium, middle layer and tapetum. Next, coming to the inner spore mother cell. Spore mother cells takes biotic division and produce haploid microspores which are called pollen grains. This is about development of anther or microsporangia. Next, coming to the our main topic, structure of anther. Anther wall consists of 4 layers. They are epidermis and endothelium, middle layers and tapetum. Microsporangium or anther wall consists of 4 layers. They are epidermis, this pink color, epidermis and endothelium, middle layer, tapetum. Epidermis anther wall consists of 4 layers. They are epidermis, endothelium, next middle layers and tapetum. Coming to the epidermis. This is epidermis. Epidermis is outermost layer, one cell thick without any intercellular spaces. It is a protective layer. Epidermis is the outermost layer, one cell thick without any intercellular spaces. It is a protective layer. The epidermal cells in mature anther become stationed and much flattened. Some of the epidermal cells are bigger in size. These bigger size cells are called stomium. Bigger size cells are called stomium. These are the stomium cells. Stomium cells. Coming to the anther epidermis. Epidermis is the outermost layer, one cell thick without any intercellular spaces. It is a protective layer. The epidermal cells in mature anther become stretched and much flattened. Some of the epidermal cells are larger in size and it is called stomium. Larger size cells are called stomium. The stomium helps in the dehiscence of anther. The stomium helps in the dehiscence of anther. Next, beneath the epidermis, endothelium is present. The cells are large in size and do not have intercellular spaces. These cells are made up of cellulose and alpha-cellulose. Next, beneath the or below the epidermis, endodermis is present. These cells are large in size and do not having intercellular spaces. These cells are made up of cellulose and alpha cellulose. Next, coming to the middle layers. Beneath the endothelium, middle layers are present. It is made up of 2 to 4 layers of beneath the endothelium. Middle layer is present. It contains two to 3 layers. The cells of the middle layer filled with starch grains. The cells of the middle layer filled with starch grains. Beneath the endothelium, middle layers are present. It contains 2 to 3 layers. The cells of the middle layers filled with starch grains. Middle layers degenerate by the time of meiosis takes place in the microspore mother cells. Middle layers degenerate by the time meiosis takes place in the microspore mother cells. This is about middle layers. Next, coming to the tapetum. The tapetum is the innermost layer of the anther wall. It is surrounds the microsporogenesis tissue. This tapetum provides nutrient food material to the microspore mother cells. Based on the development, tapetum is divided into two types. They are amoeboid tapetum and second one is secretory or granular tapetum. Next, coming to beneath the middle layers, tapetum is present. It is the innermost layer of the anther wall. It is surrounded the microsporogenous tissue. This tapetum cells provides the food material to the microsporogenous tissue or microspores. The tapetum cells having dense cytoplasm and a large nucleus. Based on the development of tapetum is divided into two types. They are amoeboid tapetum and secretory or granular tapetum. Once again, we discuss about epidermis, endothelium, middle layers and tapetum. Epidermis, it is the outermost layer, one cell thick without any intercellular spaces. It is a protective layer. This epidermis is the outermost layer, one cell thick without any intercellular spaces. It is a protective layer. The epidermal cells in a mature anther become stretched and much flattened. Some of the epidermal cells are large in size. They are called stomium. The stomium helps in the dehiscence of anther. Next, coming to the endothelium. Beneath the epidermis, endothelium is present. The endothelium cells are large in size and do not having intercellular spaces. These cells are made up of cellulose and alpha cellulose. Next, coming to the middle layers. Beneath the endothelium, middle layers are present. Middle layer wall contains 2 to 3 or 2 to 4 layers. These cells are filled with starch grains. The middle layer cells are filled with starch grains. The layers degenerate by the time of meiosis takes place in the microspore mother cells. Next, coming to the tapetum. Tapetum is the innermost layer of anther wall. Beneath the middle layers, tapetum is present. This tapetum surrounds the microsporogenous tissue. This tapetum provides the nutrients or food material to the microspores. The tapetal cells have dense cytoplasm and large nucleus. Based on the development of tapetum, this is two types. This tapetum is two types. They are amoeboid tapetum. Second one is secretory or granular tapetum. Coming to the amoeboid tapetum. Amoeboid cells are formed from immature tapetal cells. Amoeboid cells are formed from immature tapetal cells. Example of the amoeboid tapetum: lower dicotyledons and lower monocotyledons. Next, secretory or granular tapetum. Secretory tapetal cells develop from matured cells of tapetum. Many dicotyledons and higher monocotyledons we can see secretory or granular tapetum. Next, coming to the microspore or pollen grains. The microspore mother cells takes meiotic division and produce pollen microspore tetrads or pollen grain tetrads. The microspore or pollen grain is uninucleate, single-celled, uninucleate and globular. The microspore mother cells takes meiotic division and produce microspore or pollen grain tetrads. The microspores or pollen grains are single-celled, uninucleate and globular. The outer layer of the microspore is made up of pectin and inner layer of the pollen grain is made up of cellulose. The outer layer consists of sporopollenin, which checks the natural decay of the pollen grains. It is due to the sporopollenin that the pollen grains get preserved during fossilization. The outer layer has pollen kit. It produces a oily coating over some oily coating. This oily coating pollen is called pollen kit. It helps as an insect attractant during the pollination and protect pollen grains from UV radiation. Microspore mother cells takes meiotic division and produce haploid microspore or pollen tetrads. The microspore or pollen grains single-celled, uninucleate and globular. The outer layer of the pollen grain is made up of pectin. The outer layer is called exine. Then inner layer is called intine. Outer layer is made up of pectin and inner layer is made up of cellulose. The outer layer consists of sporopollenin, which checks the natural decay of the pollen grains. It is due to the sporopollenin that the pollen grains get preserved during fossilization. The outer layer also contains pollen kit. It is a oily coating. It helps as an insect attractant during the pollination and protect pollen from UV radiation. Pollen kit helps attracting insects and protects pollen from UV radiation. This is about anther structure or anther structure, microsporangium structure. Next, In next video or next class we can learn microsporogenesis. Thank you.